Our next presenter is Doug Nelson from the College of Pharmacy, the Pharmaceutics PhD program.
Do I hit this? Alzheimer's disease is a degenerative brain condition that erodes memory, thinking, and personality. Today, more than 10% of the US population over 65, and 1/3 of those over 85 suffer from the disease. The challenge of providing compassionate care often depends on the devotion of loved ones. The social responsibility of medical science is to match this dedication of millions in our search for a cure. The fact is, more and more resources are being directed into Alzheimer's research, but effective treatment remains elusive. The removal of proteins that comprise toxic plaques in the brain is the strategy of most recent therapeutics. Unfortunately, they've shown limited ability to slow cognitive decline and come at significant risks. Another important but less studied feature of the disease is the accumulation of iron in the brain. Interestingly, we find iron deposits in the same location as dense plaque formations, and the degree of iron accumulation correlates to cognitive decline. This poses two questions that my research seeks to address. How does the iron get there, and what does it do? Healthy brain function depends on the reliable delivery of nutrients from the blood to the brain and the removal of toxins from brain to blood. Tight regulation of this traffic is carried out by a vast network of specialized tiny blood vessels, often referred to as the blood-brain barrier. The structure of the blood-brain barrier is comprised of endothelial cells, which are surrounded on the outside by important supporting cells called pericytes. Not to be confused with parasites, pericytes regulate blood flow and many key functions of the blood-brain barrier. But this barrier and transport system breaks down in Alzheimer's disease, and among the first signs of this vascular dysfunction is the degeneration of pericytes. We have found that pericytes control expression of genes in the endothelial cells which regulate iron trafficking. These findings predict that pericyte loss opens the gates to iron accumulation in the brain. The problem with excess iron is that it causes ferroptosis, which is a cascade of events that leads to cell death and cognitive decline. Alzheimer's is a complex disease, likely requiring multiple strategies in order to effectively treat. Strategies we intend to investigate include the removal of toxic iron, protection of pericytes, and the repurposing of existing drugs to improve vascular function. This and other avenues of current Alzheimer's research are all very important because each piece of the puzzle brings us closer to a cure. Thank you.